Hey guys, it's Mrs. Ullery, and in this video I'm going to talk to you about the macromolecule group nucleic acids. So there are two main types of nucleic acids. There's DNA, which holds the instructions for building protein, and then RNA that helps read the DNA instructions and translate these instructions during protein synthesis. DNA stands for deoxyribonucleic acid. And again, it is the molecule that carries instructions for life, specifically making proteins, which then build life and regulate all of the processes of life. This genetic information is encoded in the sequence of 4 nitrogen bases, so A's, T's, G's, and C's. And the order of these nitrogen bases will determine the genetic code. DNA is also described as a double helix, meaning that it has 2 sides to the strand and that it is a twisted shape. So, DNA is a polymer made of repeating monomers. And so each of these monomers are called a nucleotide. And a nucleotide has 3 parts to it. They have a phosphate group, they have a 5-carbon sugar, and then they have what's called a nitrogenous base, or a nitrogen base. And it's the nitrogen base that gives the nucleotide its identity. There are 4 different types of nucleotides in DNA. There's adenine, abbreviated as A, thymine, T, cytosine, C, and guanine, G. And these, if you look at the nitrogen bases, there's kind of 2 categories of nitrogen base that you can have. If it's a single ring, like cytosine and thymine, they're called pyrimidines. If they have a double nitrogen ring, like we see here with adenine and guanine, they're called purines. And the way to remember this, which one is which, is this little phrase, pure as gold. So purines are adenine and guanine. So now if we switch to this molecular structure of our nucleotide, instead of our generic cartoon one, we can actually see the elements that are in our nucleotides and our nucleic acids. So nucleic acids, we can see that there's a phosphate group, which contains phosphorus, a 5-carbon sugar, and then our nitrogen base. So the elements that we see in a nucleotide are C, H, O, N, and P. And then when we look at a nucleotide, it's also important to recognize that there's directionality to a nucleotide. And so to determine what the directionality of a nucleotide is, we have to look at the carbons. So we have to number the carbons. And the way that we do that is the top of the sugar, that point of the pentagon is oxygen, and think of that as 0. Then we count the carbons going around in a clockwise order. So think of oxygen as 0, and then this would be carbon 1, this would be carbon 2, carbon 3, carbon 4, and carbon 5. And remember, at each of those points on our sugar is a hidden carbon that we're not showing in the diagram. So as we go around, we can see that the 5 carbon is on top and the 3 carbon is on the bottom. A way to remember which carbon is the 5' carbon, that's the one that has the phosphorus attached to it. And then the 3' carbon is the one that has the hydroxide attached to it within the sugar. I personally just like counting my carbons and looking at the oxygen and thinking of that as 0, and then counting clockwise going around our sugar ring to figure out the order. So this particular nucleotide we're looking at is in what we call the 5' to 3' direction. And this is really important in terms of the synthesis of DNA and RNA. So if we look at this molecule here, so here's our DNA molecule, which end, A or B, do you think is 3'? Pause the video and see if you can figure it out. Okay, so as we look at it, side B has the 3' facing up, because if we look at that sugar pentagon, the oxygen is pointing down, and if we count around, we'd have our 1, 2, 3, so the 3rd carbon is facing up and the 5 carbon is facing down. On the other side, we can see that the point of the pentagon is facing up. Our phosphate is pointing up. So if the phosphate's pointing up, that means that the 5' end is up. And the opposite end would be 3' and the other 5. So on the left, we have our DNA running in a 5' to 3' direction. On the right, we have it running in a 3' to 5' direction. So let's talk about how we build DNA. So we can join these nucleotide monomers into a single chain, which you will see with RNA, but in DNA it is double-stranded. So how do we get the other side? So if we take a look, the way that we join the two sides of our molecule together is through interactions between the nitrogen bases. So A pairs with T and C pairs with G. But if we were to try and take a nucleotide and pair it up with thymine, you would see that the sugar and the phosphate would be in the way. So in order for this to happen, we have to flip our nucleotide over so that the nitrogen bases are facing each other. So A pairs with T and G pairs with C. Then to join our nucleotides together, we can see those red lines there. Those are going to be covalent bonds. So the phosphate of one nucleotide is going to join to the sugar of the next nucleotide. And because it's a covalent bond, this is going to happen by dehydration synthesis. So we keep adding our next nucleotide. We have the complement between the nitrogen bases, and then we have the phosphate covalently bonding to our sugar of our next nucleotide until we've built the chain. So each of those red lines there represent a covalent bond that's going to be created by dehydration synthesis. The outside of our double-stranded DNA, we call that the sugar phosphate backbone for obvious reasons. Because if we take a look, our backbone is made of alternating phosphate, sugar along the side. So sugar, phosphate, all joined together by covalent bonds. At the center of our double helix, or the center of our double-stranded DNA, we have hydrogen bonds. So our adenine and thymine are going to be attracted to each other through hydrogen bonding. So adenine and thymine make 2 hydrogen bonds, and our guanine and cytosine make 3 hydrogen bonds, holding the two sides of our DNA together. And then again, if we take a look at our sides, our sugar phosphate backbones run in opposite direction. So on the left side, we can see with the phosphate pointing up that we have that going in the 5' to 3' direction. And then on the other side, we can see that phosphate group on that first nucleotide is pointing down. So that one is going in the 3' to 5' direction. When we pair up our nucleotides together, so the nitrogen bases of our nucleotides together, we call these a base pair. So here in the middle, we have guanine and cytosine pairing together. And what you should also notice is that guanine, a purine, is going to be paired up with cytosine, a pyrimidine. We can see with our other base pair between A and T, we can see that, again, thymine, our pyrimidine, is going to be paired up with adenine, our purine. So these are what we call our base pairing rules, and because there's very specific ways that these nucleotides pair up, it gives us a pretty cool property of DNA. It's awesome because if you know one side of the DNA molecule, you know the other. So if we have a thymine here, we know across from it we're going to have an adenine. If we have a guanine, we're going to have a cytosine. If we have an adenine, a thymine, a guanine, a cytosine, a guanine. And so this means that the two sides of a DNA, the word that we use for that, we describe it as being complementary. So the complement to thymine is adenine, the complement to guanine is cytosine. And this allows us to do, or to copy our DNA. And so this is really important in replication. If we have one side of our DNA molecule, we can build the other using it as a template. The other thing to notice here is that our DNA strands are moving in opposite directions. So you can see on the left side, our phosphate is pointing up, meaning that our 5' end is up and our 3' is down. And then on the other side, we can see that it's 3' to 5'. And so because these sides run in parallel to each other, but in opposite directions, the word that we use to describe that is called anti-parallel. So parallel to each other, but in opposite directions. So our second type of nucleic acid is RNA. When we get into our protein synthesis unit, RNA will become even more important to you, but for now, know that it is a type of nucleic acid that is single-stranded. So there is not a complement to RNA. It is a single-stranded molecule. So whereas DNA is double-stranded, RNA is single-stranded. So comparing these two molecules together, the structure, DNA is double-stranded, RNA is single-stranded. When we look at the monomer, they're both nucleotides. So DNA is made out of a nucleotide, RNA is made out of a nucleotide. But what's different about these nucleotides are the nitrogen bases and the sugar that they're made out of. So when we look at the sugar for DNA, it's called deoxyribose, and then the sugar for RNA is called ribose, which is why it's called RNA. These nucleic acids are named based on their sugars. So DNA is deoxyribose because it has the sugar deoxyribose, and then RNA is called RNA because of its sugar ribose, so ribonucleic acid. And then the bases that they have, they share A, C, and G. So adenine, cytosine, and guanine we share in common with DNA, but the thymine of DNA is replaced with something called uracil, a U. So RNA, the bases are A, C, G, and U. So circling back to the very beginning, DNA holds the instructions for building proteins, and so the function of RNA is to be able to take that information of DNA and help translate it during the process of protein synthesis into a sequence of amino acids that can be joined together to make a protein. And we'll talk more about that when we get into our genetics unit, protein synthesis, and then also talking about protein macromolecules.